In this part of our program, we'll discuss the topic of heredity. In our discussion, we'll include topics about DNA, genes, the nucleus of the cell, and a special process called genetic determinism. But before we begin our study of heredity, we must go back and recognize that science builds its story on physics, and at the time, Newtonian physics is what was controlling the belief of science. The reason I bring this up is that if we go back to Isaac Newton, we recognize that he divided the universe into two realms, a physical realm and an energetic realm. The physical realm is expressed as matter, the energetic realm is invisible. He said that the two realms are separate from each other. Whatever is made out of matter can only be affected by another form of matter. So when scientists want to understand the nature of how life works, the first thing they recognize is that it has to be something physical because it controls a physical body. Well, there was no idea of what was controlling life back in the 1700s at all. Matter of fact, it was just a hypothesis that there were structures inside a cell called gemmules. Now, this is the belief that Darwin started with when he was talking about his theory of evolution. Gemmules were supposed to be structures that controlled the character of the body wherever the gemmules were found. So, for example, there was a gemmule for a shoulder, and that controlled the character of the shoulder. A gemmule for a neck, that controlled the character of the neck. A gemmule for the head, that controlled the character of the head. So each part of the body had a specific gemmule. Now, they didn't know exactly what it was, so they thought it was something like paint droplets. And when Darwin started talking about the nature of evolution, he was talking about how evolution is a result of accidents or mutations in heredity. And while nobody knew what heredity was, there was the philosophy of gemmules. And in that philosophy, it is also noted that gemmules, again, were like paint droplets, meaning, let's say one parent had a white gemmule and the other parent had a red gemmule. What that meant is when they would mate, the offspring would have a mixture of red and white coming together like paint, making a pink offspring. So the idea that traits would be passed on to the next generation and that the next generation would share their traits with their parents. So the idea fit very nicely, but the question of what gemmules were, nobody had an idea as to what they were. But interestingly enough, at around the exact same time that Darwin was doing his work, there was another researcher in the field who was also looking at heredity, and this researcher was the one who started to put together the results of mating experiments. Now, you may have heard the name Gregor Mendel. He was a monk, and as a monk he was raising plants and breeding them and looking at the results of the breeding process. What he did in his study was he started to find out that if he bred a red and a white flower, that in fact the offspring were pink. And that fit the story of gemmules, that the paint drops of the color came together, so the red flower and the white flower became a pink flower. But something unusual happened, and that is when he mated two pink flowers and mated them together. In fact, what came out were three different kinds of flowers. There was a white flower, a red flower, and of course a pink flower. The point is very important. It says that the hereditary things, be them gemmules or whatever they were, did not blend with each other, that the hereditary factors were separate independent elements that come together, such as the red and the white one coming together making a pink flower, and then the result is if you mate the pink flowers, the red and white genes could be separated again and come back out. So basically it said whatever is controlling heredity were distinct characteristics that did not blend. They were units of information. However, at this point, there was still no idea of what these units of information represented. But there was another discovery that came around the end of the 1800s. In the 1890s, a scientist by the name of August Weismann was looking at cells in a microscope. And what he noted is that when cells are ready to divide, before they divide, inside the center of the cell, the structures of thread-like things were found, and these thread-like things divided before the cell divided, and when the cell split into two cells, half of the threads went into one cell and half of the threads went into the other cell. So August Weismann, looking at these threads, which are called chromosomes, said that these were the physical elements that were connected with heredity because they doubled before the cell divided, and then each of the sets of these chromosomes split up, and one set went into one daughter cell and one set went into the other daughter cell. So when putting all these things together, a new theory of heredity emerged, and that theory is chromosomes inside the cell carry hereditary information, and that this hereditary information is passed via these structures. Well, the problem is, what are the structures made out of? So finally, they started to look, and guess what they found? That the chromosomes were made out of 50% DNA and 50% protein. Now the question is, which part carries the heredity, the DNA or the protein part? Well, that question was finally resolved in 1944. And in this time, there was an experiment, a very famous experiment, Avery, McLeod, and McCarty. And here's what they did. There were two types of bacteria, physically different. One type of bacteria, type R, it makes a little point-like colony, a little tiny round dot-like colony. Type S, another bacteria species, made a big puddle-like colony. So there were two different kinds of colonies. Type R, little spots, and type S, big puddles. So here's what they did. They started growing type R in tissue culture, so they have a culture dish with type R bacteria in it, and they formed these little colonies. But what they did is they took the chromosome from the type S bacterium, and they split the chromosome into DNA and into RNA, the two parts that make up a chromosome. And in part one, they put the protein from the chromosome into the culture dish. So what they were mixing was protein from chromosome S, the puddle colony, mixing it in with the cells type R in the tissue culture dish. But you know what happened? Nothing. All the colonies stayed as point-like little round colonies like type R. So the S protein from the chromosome did not do anything. However, when they took the DNA from the S chromosome and added the DNA to the culture of R bacteria, guess what? Now there was a transformation of the R-type bacteria into S because they made big puddle colonies. The point is a very important conclusion. It says that heredity is connected to the DNA. And then what they started to find out is DNA is a very, very long molecule. It's like a long thread. And what happens is, along the thread, there are units of information. And these units, called genes, are giving the character to the offspring. So they started to find out that DNA was a hereditary material in the chromosome, that this DNA was broken up into different segments, and each segment controlled a different character, and so each segment is a gene. So a DNA is a long filament molecule, and a gene is just a segment of that. So there's a whole sequence of genes down the length of a DNA molecule. So when we look at a DNA molecule, it's important to understand, well, how does it work? Well, interesting enough, in 1953, the answer came. And the answer came in the form that DNA was a double helix. Well, this research is given credit to Jim Watson and Francis Crick. These are the men who are credited with discovering the DNA double helix. But this is a time to adjust history a little bit, because the real discoverer of the DNA double helix was a woman by the name of Rosalind Franklin. And her research was given away to Watson and Crick without her even knowing they did it. So Watson and Crick were studying Rosalind Franklin's research, and she didn't even know they were doing it, and she had the results of a DNA double helix, and they announced the results before she did. The problem is this. The real discovery of the DNA double helix is not Francis Crick and Jim Watson. It's actually Rosalind Franklin. So I say, but what did we see with the DNA double helix? And what we saw is a very important story. That these are long polymers. That means long molecules made of repetitive units, like consider a chain of pearls, and that each pearl is a unit of a long molecule. The whole molecule is called DNA, but each pearl is called a base. And there are 4 different bases that make up DNA. So as you go down the length of the DNA, the steps that we see between the 2 strands of DNA are made out of bases. The 4 bases are called cytosine and guanine, adenine, and thymine. What's very important about this is the 2 bases bind to each other. Cytosine binds to guanine, guanine binds to cytosine. Adenine binds to thymine, and thymine binds to adenine. You say, so what's the importance of this? And here's the answer. If I have one strand of DNA and a sequence of the bases along on one side, it would automatically mean that another chain of DNA will complement the original chain. So where there's an adenine in chain 1, there's going to be a thymine in chain 2. Where there's a cytosine in strand 1, there's going to be a guanine in strand 2. The point is very important. It says that the sequence of the bases along the DNA makes a replica in the opposite chain. So the 2 strands of DNA have complementary pairs of nucleotide bases, the pearls that go up and down. So if you have one strand of DNA, you automatically know what the complementary strand is going to be, because wherever there's an adenine in strand 1, there's a thymine in strand 2. Where there's a thymine in strand 1, there's an adenine in strand 2, etc. So DNA represents 2 filaments of DNA twisted together in a helix, but the steps between the helix are the bases, and they're in pairs. Cytosine bonding to guanine, adenine bonding to thymine. And so basically there's a code along the DNA. And when they wanted to understand how it worked, they saw that the code in the DNA is what provided the sequence of amino acids that make up the protein molecules. Now remember, the protein molecules give the structure and the function to the cell. But proteins wear out. Proteins break down. So for example, if you put a piece of meat on a plate and come back a few days later, the protein will be all broken down into mush. So because protein breaks down, that's what they were looking for in regard to heredity, is what is going to replace the protein. If a protein breaks down, how am I going to get another protein? The answer is heredity. I said, yeah, but where's that come from? I say, the DNA is a code that determines the sequence of amino acids in the protein. So the DNA codes for the protein. So the structure of the body, the character of the body, is determined by the proteins, and the DNA is the code that makes the protein. Now the question that scientists had to face is, well, DNA makes protein, protein gives us our behavior and our structure. The question is, what controls DNA? Well, they did an experiment, and this video shows that experiment. You start with a DNA double helix, and then you remove one of the helices, so now there's only a single strand. But you take that single strand of DNA and put it into a beaker where there's a solution of the building blocks, the adenine, cytosine, guanine, thymine, these building blocks, you have a solution of them. But when you stick a single strand of DNA into it and incubate it, when you pull out the strand, guess what? It's a double strand. It copied itself. That was the big answer they were looking for. They said DNA controls the protein, but what controls the DNA? And this experiment said the DNA controls itself because it copies itself. There was one big problem, and this problem changes the world, and that is this. When they did this experiment, they did this with pure DNA. There was no protein from the chromosome involved. I say, why is it relevant? Because in the cell, there is no such thing as pure DNA. DNA is always connected with protein. But in these experiments, they didn't even use the protein. They just used the pure DNA, got the results that the DNA copied itself, and what is the result of that? And it means this: DNA reproduces its own self. DNA reproduces the proteins. So if you want to see what controls biology, you just have to go to the DNA. There's nothing above the DNA because the DNA can do its own recording. the solution came, and the solution was actually described in a hypothesis referred to as the Central Dogma. The Central Dogma is a hypothesis that was made by Francis Crick. And in that hypothesis, this is what Crick said. Information in biology flows just as we just said. It starts with the DNA, it then goes to the protein, and then the protein gives us the behavior and the structure. So basically it says information flows from DNA to the protein. But in between, they found there was like a molecule, like a copy of the DNA, like a photocopy of the DNA. So the original molecule is called the DNA, but from the DNA there's a copy made called RNA. It's a different version of nucleic acid. It's just like a blueprint copy of the original gene. And that the RNA molecule is actually used to make the protein. So the point is simple. To make a body, information starts in the DNA. It is translated into RNA, a second kind of molecule, and the RNA molecule is the actual template that builds the protein. And this is what is described as the flow of information in biology, and the official title of this hypothesis is called the Central Dogma. And it is the foundational belief of biology up until recent times. And the why I made the slide look like the Ten Commandments is the Central Dogma is like the Ten Commandments of biology. That information flows from DNA to RNA to protein. We are the protein. Well, there's a very important conclusion about this. It says that genes control life, yeah, but the fact is, if you look at the message information, it only goes one way, from the DNA to the RNA, and from the RNA to the protein, only one way. And you say, why is that important? And the answer is very important, because it says, since we are the protein, can we change our DNA? According to this hypothesis, the answer is no, because information cannot go backwards. So we're left with a final conclusion of the Central Dogma, and that is genes control life. And it is based on this that we teach this in a medical school for years. Now, I said it has been recently changed by science, but most people in the world still have the old belief. And I say, well, what is the belief? Well, think about it this way. What determines the character of our biology and our behavior and our emotions? And the answer, according to science, is genes control this. This is a belief called genetic determinism. This is what the public believes. Now remember, leading edge science does not believe this anymore, but the public still believes it. And I say, well, what is the belief? Well, the belief is genes control the character of my life, genetic determinism. My life is determined by my genes. I go, why is this relevant? And the answer is, well, if you ask somebody, did you pick the genes you have? The answer is, not that I know. And they say, well, if you don't like the characteristics, can you change your genes? And the answer is, no. And I say, and the belief is the genes turn on and off by themselves. Yep, self-control. I said, well, what does that mean? I said, that means that we are victims of our heredity, that the genes that we got from our parents come into our body, give us the character of our body, our behavior, our emotions, and that we don't control the genes, they control us, so we're victims of our genes. And people walk around thinking that all their illnesses and all their problems in life come from the genes. Well, important point about this is very simple, and the point that I need to make is this. Do the genes control life? Well, now let's go back into the cell and say, where are the genes? Well, about 98% of all the genes in the cell are present in a large structure, an organelle called the nucleus of the cell. And since the genes were supposed to be able to turn on and off by themselves and control biology, then the genes in the nucleus are actually then controlling our own biology. And therefore, that's where we bought the belief that's in the textbooks right now. If you go to a school and read a textbook, it says that the nucleus of the cell, that's where the genes are, is the equal part of the brain of the cell, because that's where the decisions are made. Well, is this a true statement? Well, let's look at it this way. if I remove the brain from any living organism and throw it away, what is the consequence? What is the result of doing that? And the answer is, the organism dies. I go, yeah, if you take the brain out of an organism, lost the control, the organism's going to die. So I say, good. But now I go to a cell. And I say, what if I remove the nucleus of the cell? Because the nucleus is supposed to be the brain. What would happen then? Well, this is a process called enucleation. It's been done a lot. And I go, well, why is it relevant? Because I say, what happens when you remove the nucleus from the cell? And the answer is this. The cell stays alive for months, and the behavior is totally unaffected. It still has all the complex life behaviors. Digestion, respiration, excretion, motility, the ability to communicate with other cells. All of the functions are still there, even though the cell has no genes. Well, the point is very important, and the point is simply this. Genes are not necessary to control biology. The cells work without genes in them. So the most important point is this. The nucleus can't be the brain of the cell because the cell can live without the nucleus. So we go back and say, well, we thought the nucleus was the brain of the cell because that's where the genes are and they control life. But I say, then what is the function of the nucleus? It's the biggest organelle in the cell. What is its function? And the answer is simply this. The function of the nucleus is that it's used to reproduce the proteins. Remember, proteins break down. I have to replace them. So the nucleus is used to replace the proteins, and the nucleus is used to reproduce the whole cell. The joke that's very funny is the nucleus is not the brain of the cell. The nucleus is the gonad of the cell. It's the reproductive element of the cell. So our belief that the nucleus is the brain of the cell is completely wrong. It's only used for reproduction. It doesn't tell the cell what to do and how to do it and where to do it. That information does not come from the nucleus. And so we have to really look at another understanding. So let's first go back and say, so tell me the story of DNA. I say, well, remember a story about proteins. The proteins of our body give us our structure and our functions. Each protein, there are 100,000 different ones, each protein has a backbone made out of amino acids. And the relevance is the backbone gives shape to the protein, and therefore the structure of the protein is due to the backbone. But the backbone is a sequence of amino acids. And there are 20 different kinds of amino acids, so as you assemble a backbone of a protein, you can create many different combinations. But each different protein has its own special sequence of amino acids. So the blueprint of a cell is the blueprint of the protein. I say, yeah, but what gives me the structure of the protein, the blueprint? I say, the nucleic acid. The nucleic acid has those bases, the adenine, thymine, cytosine, guanine bases along the length of the molecule. And the sequence of the bases is a code to tell which amino acid comes after which amino acid comes after which amino acid in creating the backbone of a protein. So the point is simply this. Nucleic acid is actually a blueprint. And you say, why is that relevant? So I show you this picture of a blueprint and ask the question, is this blueprint on or off? And if you said that to an architect, she would probably laugh at you because she says it's a blueprint. There's no on and off to a blueprint. And I go, this is the most important point of biology at this moment, and that is this. For years, people have said genes turn on and off. A cancer gene turned on and caused cancer. A gene caused a cardiac disease. A gene causes this. The fact is this. Genes do not control biology. Genes are blueprints. They cannot turn on. They cannot turn off. They're information. And the point that we really have to understand about this is, how come we keep talking about genes turning on and off, controlling life? And I go, because there's an error in the language. Biologists have mixed up two different words and made them the same when they're not the same. The two words are causation and correlation. In English, causation means the act or agency which produces an effect. Correlation in English means something that is related to or connected to something. I say, why is it relevant? The two words are not the same. Genes are indeed correlated with diseases. Yeah, there's a gene correlated with cancer. There are genes correlated with Alzheimer's and all that. But the most important point is this. The gene did not cause the disease. This becomes very, very important because people believe when they have a certain gene, they're automatically going to have a certain disease. So, for example, a number of women are told they have the BRCA1 cancer gene and immediately feel they have to have a mastectomy to remove their breast so they don't get the cancer because they have the gene. The point is very, very important. The gene does not cause cancer. There is no such thing as a cancer gene. There is no gene that causes cancer, period. The gene is correlated with cancer, meaning this. The cancer didn't come from the gene. The cancer is derived from a lifestyle that activates this gene. If you have a healthy lifestyle, you can have this gene and it'll never affect you. As a matter of fact, 50% of the women that have the BRCA1 gene never get the cancer. So the most important conclusion right there is having the gene did not cause the cancer. And then I say, yeah, but 50% of the women did get cancer. I say, and how did that happen? I say, not because they had the gene. It's because they were not living in harmony with life. And that the stresses they were facing were things that activated the cancer gene. So it becomes very important for us to understand a conclusion. Genes do not control our biology. Genes are blueprints to make the proteins that give us the behavior. But that is not controlled by the gene. The gene, as I said, being a blueprint, does not have on and off. It's controlled by the nervous system. Genes are controlled by the nervous system responding to the environment. And as a result, as the environment changes, we also change our genetic activity. So having a gene does not mean this is the whole rest of your life, because the genes can change their activity based on how we respond to the world around us. So the belief in genetic determinism, the belief that genes can tell you about the future, is completely false. In fact, there are only six diseases in large numbers that are caused by a gene. And these are the following diseases as they roll across your screen. And I go, why is this important? And the answer is this. People are looking at getting their genes read. And I say, well, what good is having a gene readout? And I'll say there's one good thing. Number one, you can find out your lineage. You can find out where your parents came from. And you can find out if you're a carrier of one of those 6 gene diseases. But if you don't have those 6 single gene diseases in your family, then it's not even worth doing the genetic readout for the following reason. If you want to predict the future by reading your genes, that's not possible. And the reason is this, as we'll discuss in the next section. The activity of the gene changes in regard to how we respond to the world. What we believe, what we perceive, the environment we live in are all factors that can change your genes. And therefore, you can show me a gene readout and I go, so what? The reason is this. It's really based on the life you're going to lead. That will determine the activity of those genes. So, conclusion is very important. The belief that genes control our lives is completely false. With the exception of the 6 single gene diseases I mentioned, all other diseases are caused by complexes of genes, large numbers of genes. For cancer, for example, 14 genes just to get the cancer off the ground, and 200 genes are involved to create the actual cancer. So the idea that there's a single gene that causes cancer is a totally false assumption. That diseases are actually caused by combinations of genes. And in fact, a new finding is also important. Less than 1% of all diseases are connected to genes. Over 90% of all disease has to do with environment, lifestyle, and belief. And we'll go into that deeper in the next section.
The World Summit of Integrative Medicine. Sponsored by Quantum University.
Hi, dear friends and cultural creatives. Bruce Lipton here. Today I have a wonderful program on the new biology and the healthcare revolution. This is science based on the research that I did while I was teaching at the University of Wisconsin School of Medicine and later doing some pioneering research on stem cells at Stanford University. This is an introduction of how we can connect the energy medicine to the conventional biology that we've read about in schools. When Isaac Newton was looking at the nature of the universe and the movement of the planets and the stars, he invoked the idea of Descartes that the universe was like a giant machine. And the significance is that in his data, the only thing he offered in the equation were physical characteristics of the planets and their movement, no invisible energetic characteristics. So the science that Newton was working on was a science based on the physical material world. So the science that evolved from Newton has been called scientific materialism. And scientific materialism means if you want to understand the nature of the universe and everything in it, such as us, then you don't have to look for invisible forces. You just study the physical realm. Mind and the invisible forces were eliminated from understanding the nature of the human body. But when we look at the human body in a Newtonian perspective, then we see the human body as a machine. It's a physical element that can be taken apart and the pieces studied. So scientists started to take the body apart. First there was gross anatomy, where they just started to look at what you could see with your eyes only. Later, when the microscope evolved, then they discovered what is called microscopic anatomy, where you then looked at those structures like the muscles and the organs and the glands, but you now looked at them with a microscope and looked at the cells and the tissues. And then after that, a new field came in, which is called biochemistry, which then further took the machine apart down to smaller and smaller pieces. Well, the final taking apart of the body really occurred in 1953, and that's when the double helix image offered by Watson and Crick ended the search for how biology worked. Because once the double helix was found and the DNA was found, science said, oh, now we have an understanding of how the biology works. There's a physical thing called DNA, and the physical DNA programs the nature of the body and its behavior. So we bought into the story that DNA is actually the source of our physical structure and our behavioral characteristics. Since the DNA is believed to control the biology, then the nucleus is like the cell's brain, that the DNA in the nucleus controls the behavior and development and functions of the cell. And I said, well, is this really true? Is the nucleus the brain of the cell? Well, the relevance about that is then very simple, and it goes like this. It says that if you remove the brain from an organism, then what is the consequence of that action? What happens when you remove the brain from an organism? And the answer is straightforward and simple. It basically says that the organism dies. And so that without a brain, there's no control and the organism dies. And I said, well, okay, let's go to a cell. And in that cell, we say the nucleus is the brain of the cell. And then I say, well, if I remove the nucleus, like removing the brain, the cell should die. And it's a process called enucleation. And enucleation is where you go into the cell with a micropipette and you pull out the nucleus of the cell, so the cell has no genes in it anymore. And I say, well, what's the consequence of doing that? Does the cell die? And the answer is absolutely not. The cell doesn't die. The cell lives, and its behavior is exactly the same. It's unaffected, and the cell can live for months without the DNA. I said, well, why is that relevant? And a simple point is this, well, then the DNA is not controlling the biology of the cell. The nucleus cannot be equal to the brain of the cell. And so now we're left with a very interesting challenge, because we say, well, the nucleus is controlling everything in the brain, and that led us to believe what? That led us to believe that DNA makes decisions. So all of a sudden it says, wait, the whole biology you were brought up with, that genes control life, turns out to be totally wrong. So let's go into a new biology. Let's talk about something new, but we'll first give a big fact conclusion. This is an absolute fact. Genes do not control biology. That is a fact. There's a new story we've touched upon in the past, a story about something called exosomes. Exosomes are little pieces of cells that pinch off as little tiny vesicles. It's so small you can't see them in a regular microscope actually easily. And these exosomes were thought to be degradation products. In other words, a cell was removing a piece of the membrane and cytoplasm, and so that it would be broken down and the building blocks recycled. So exosomes originally thought were just a way to remove parts of the cell that need to be replaced. But as I started to notice, many of these exosomes were not degradation particles, but they actually contained information, DNA, RNA, proteins that would affect a cell if that exosome was picked up by another cell. So cell A releases an exosome with information that controls the genetics of a target cell. Most important in regard to cancer that we're just talking about, a cancer cell can arise anywhere in the body. That's a fact. Could arise anywhere. But there are very preferred sites where cancer grows, such as the liver and the lungs, pancreas, different sites where cancer seemed to be manifesting. And our original thought was, oh, the cancer was created in that organ. Oh, the cancer formed in the lung, or the cancer formed in the liver. Well, there's a whole new story. Exosomes are vesicles that pinch off a cell containing molecules that control the behavior of a cell that will pick it up, controlling the genetics and the protein synthesis and all that. So by definition, an exosome is a virus. It carries a piece of information from cell A to cell B. And cell B isn't random, meaning on the surface of the exosome, which is a membrane-wrapped structure with some cytoplasm inside containing nucleic acids and control factors, such as things that control the immune system, that these exosomes are information carrying structures that are picked up by a cell. The cell that picks it up, again, is not an accident, because on the surface of those exosomes are receptors that complement specific cell types. So let's say a precancerous cell is forming in the leg. I go, then what? Well, we now know that those cells release exosomes. I go, yeah, but this information carrying vesicle, what does it have? Well, first of all, it's directed to a site, the liver, the lung, the pancreas. So that the cell releases these information containing virus particles, I'm going to just give it a name, virus, that is sent to a specific site, and the cells at that site pick up these viruses, and those cells then change their function based on what information was in that exosome. And all of a sudden I said, oh my God, it's called terraforming. In other words, the precancer cell releases the exosomes. The exosomes go to the site where the cancer will grow, which is not where the precursor cell is in the first place. As I said, precursor cell could be in the leg, but the exosome is directed to cells in the liver. When the exosome gets to the liver and the cells pick it up, they change their function to create an environment, terraforming, that will support the cancer cell, which is going to travel there in the future. Oh my God, a cell in some place in the body sends out a signal to a destination, such as liver or lung, and says, prepare the site, because this cell is going to move from where it is now. It's going to go through the system and end up in the liver or the lung or that destination where the exosomes were. So that the exosomes carry information to another part of the body to prepare for a cancer. And all of a sudden I go, wow, then these exosomes are not just degradation things. No, these are information containing viruses that come from a cell source, but have a very specific destination. It's not accident or chance. It's like, oh my God, we never understood anything like this. We never knew our own cells are communicating at another level, because we think in the body, communication is nervous system is communicating, but nervous system only actually contacts a very small percentage of the 50 trillion cells in your body. So not all cells are directly connected to the nervous system. Another way of communicating with the 50 trillion cells is to release hormones or, neurofactors that go in the blood.
And I go, well, that's really interesting, because that is a way of coordinating the body, because the blood is going to travel through the body carrying this information, and the cells that are responding to the information are going to adjust their behavior based on what the hormones are. Yet there's a problem. And the problem is this: there are two very important messages that cannot be trusted to the nervous system, because not enough cells are connected to it. And it can surely not be connected to hormones, because all cells are going to be exposed to it. And I said, these two messages are the message to reproduce more, or the message to die. If those messages go to the wrong place, the whole system could collapse. So I said, well, how do you assure that a message to reproduce is going to go to the right place, or a message to die is going to go to the right cell? And the answer is, exosomes on the surface have these receptors that determine the destination and which cell is going to pick it up. So sending exosomes is one of the highest levels of communication. It's like a zip-coded telegram that is being sent. Now back up. Exosomes are in the blood. They're all over the place. How many exosomes? If I took a blood sample from every one of you right now, I said, let's count the exosomes in your blood. Here's how many are going to be in there for the average person. 2,000 trillion. Okay, that's a big number. Okay, let's put it in another word. 2 quadrillion. Well, that's, I mean, millions, billions, trillions, quadrillions. That's a number that's mind-boggling. Right now, every one of us has 2 quadrillion exosomes in our body. But guess what? When a person has cancer, they have 4 quadrillion exosomes in the blood. What's the point? You can see a person is going to develop cancer by counting the population of exosomes. If they go above the 2 quadrillion and start going to 3 or 4 quadrillion, that's an internal message system that says that a cancer is evolving. So guess what? Now we can even do a survey of how many of these exosomes are present in a body, which is indicative of a future cancer unfolding. So, oh my God, there's a test that can let you know if you're moving toward cancer. So the whole idea about that is simply this. We go back to the family with the gastro problem where the stomach creates a cancer. I say, before you do anything radical like cut the stomach out, there are 2 things that we should be doing. Number 1, assessing what is the common psychology of those in the family that get the stomach cancer versus those 25% that don't get the stomach cancer, because there's a psychological input to this. It's not an accident. Cancer is not an accident. It is well choreographed, well planned. So we can start to say, well, what psychology is behind those that end up having the cancer? And number 2, before you do anything radical, you can do a survey of the exosomes. And therefore, before you cut somebody's stomach out, you can see, are they moving toward a cancer or not by looking at that population. I bring up exosomes because this is brand new in the sense of meaning of what's relevant to our lives, to recognize that our own cells create viruses. Now, the problem with the word virus, as soon as you say the word virus, a lot of people get like a stomach tightens up and go, horrible, viruses are scary, viruses cause all the problems. And it's important to note, very important, biology doesn't create something that's all bad. Well, we focus on viruses being bad. We might have to ask, is there a good point to viruses? And the answer is absolutely. Because viruses are communications that are so specific, they can be sent to specific cells out of 50 trillion. You can target a very specific cell type if you have a virus with a zip code receptor system that only identifies that cell type. What's the point? It is the most secure form of communication in a biological system, exosomes. They're new. Their understanding is just coming into the world. I need you to understand this because let's stay ahead of the curve on things. Exosomes are found. Exosomes coordinate the community of 50 trillion cells so they communicate with each other and create an environment that supports what's going on. Now, if it's a negative environment, then of course, then we have to deal with things like cancer and pathology. But the same viruses are very important for normal functions. I'll give you one example where this virus is so important, exosome virus, and that is this. When a woman is pregnant, recognize that the placenta and the fetus have different genetics than the mother because they represent the composite genes of the father and the mother together. So fetal cells don't have the same genetics as the mother. And I say, well, why is it relevant? Because the placenta, the cells of the placenta merge with the uterus and penetrate the uterus. Finger-like projections of placenta go deep into the uterus where they contact the mother's blood supply and open up a passageway so blood can go from the mother system into the fetus and nourish the fetus and help the development of the fetus. I go, wait a minute. By all rules, the mother should not be able to get pregnant because if the foreign cells start to invade her uterus, that's the equivalent of cancer. And by definition, the mother's immune system should reject the fetus. I go, well, obviously it doesn't. How can the mother's immune system be prevented from rejecting the foreign fetus? And here's what we now know. Exosomes. The placental cells make exosomes, particles in membranes with control information that control the immune system of the mother. Inside are special molecules that are actually hormones for the immune system. They're called cytokines. Cytokines manipulate the function of the immune system. Well, the foreign fetal cells make exosomes, virus-like particles that are picked up by the mother's immune system. And these virus-like particles redirect the immune system to go away from the placenta so that the immune system is not recognizing the foreign fetal tissue because the foreign fetal tissue sends viruses with cytokines that shunt the function of the immune system and push it to go somewhere away so that the placenta can grow and mix its cells with the mother's cells. It's even been suggested that at term, when the baby is being born, part of it is an immune rejection, that birth is an immune rejection, that the mother's system at birth pushes the placenta away, disconnects it from the mother's uterus, and in that process engages in birth. So I wanted to introduce the exosomes because I talked about them before, but they're becoming more and more a topic of discussion. And so you won't be unaware of the future of what's happening in this biology once we start to recognize that our own cells are communicating with each other through viruses called exosomes, and that these viruses are used to coordinate the function of the community. Now, that coordination could be toward the benefit of the community, but misdirected exosome coordination could lead to cancers and death. So all of you out there, when the word exosome pops up somewhere, you already are ahead of the curve because you recognize what they are. They're communications among 50 trillion cells where cells can talk to each other with specificity as much as you can send a telegram that will be delivered to a person that you specified. Only a cell can deliver an exosome to another cell that it specified. And all of a sudden we realize, oh my God, there's a higher level of communication that we hadn't even seen before. And it's so profoundly important and it's just coming into our awareness. And now you already have that information. So thanks for listening to that little caveat. So before a person in that family gets their stomach taken out, one of the easiest things to do is just do a survey of how many exosomes are running in the system. If the number goes above the 2 quadrillion, it's time to consider some kind of help to offset what is a blooming pathology. If the exosomes stay in the same normal level, then there's no necessary medical intervention involved at that point. So, new idea, old stories with cancer genes as being connected, correlated with the cancer, but not causing the cancer. I hope in some sense I shed some light as well introduce the new light of exosomes. So all you out there got to jump on everybody around you. Why? You already heard the exosomes and their positive and negative roles. The world is in a state of evolution. And what I mean by evolution is not a physical evolution. Humans are not going to change into something different. The evolution that we're really experiencing now is an evolution in consciousness. A consciousness that has in the past separated us as individuals. A consciousness that has been programmed with a belief that life is a struggle and that we're out here to survive. We have to be in competition with each other and undercut and undermine. And we were acting as individuals, looking for our own individual power. But in today's world, there's a new understanding that the evolution is an evolution of consciousness, an evolution of an expansion of we are more than just these physical entities, that there is an energy involved. In Newtonian physics, the conventional ones that most people have learned, atoms are physical particles, like little granules. But in 1925, physicists came to another understanding because they said, so what are those particles made out of you called atoms? And they went inside. And they found there were smaller so-called particles, electrons, protons, and neutrons, but that still wasn't the quest because what are they made out of? And that's when the world changed. Because when quantum physicists started to say, what is an electron or a proton or a neutron made out of? It's made out of energy, not physical, no substance. An atom is like a nano tornado, a spinning force field. There's an illusion that we are physical entities. You're an energy field. And I go, so what the heck difference does that make? And I go, well, here's the interesting part. Particles, I can separate. Here's a particle, let's talk about one particle. Energy just mixes all together. I'm sitting in a field of energy right now. So if I'm made out of energy, which I am, does it mean that the edge of my energy stops right here? And the answer is no. In quantum mechanics, I'm an energy field. Wherever I go, I'm like a broadcasting an energy. Wherever you are right now, you're broadcasting energy. And so in a Newtonian world we grew up with, this is separate from this and that's separate from this. And I go, in a quantum mechanical world, nothing is separate. Everything is an energy field with no borders to it. So what I do in my world is not just what I do in my world. What I do in my world is broadcast to everything else. And if it resonates or is in harmony with you, my energy can affect your energy. Or my energy can actually cancel your energy. Everything is interconnected. The chair I'm sitting on is energy. The air I'm in breathing with is energy. I'm energy, you're energy. We're all connected in this energy. But the fact is, energies are communications. And we vibrate and broadcast just like a radio. We're broadcasting who we are out into the field, and we alter what's going on. It is beyond language. It's a communication. It has nothing to do with words. It's an energy that's available if you just want to feel it. And I think that's the big issue. Feelings. Because feelings are telling you, you feel good? Yes, I'm having more energy. You feel bad? You feel weak? I'm having less energy. And I say, why is it relevant? It's more powerful than words. Energy can tell me, am I with people that are supporting me because I feel good around them? Or do I have a little quasi feeling of this, it doesn't feel right to me? So we have a new world that we're coming into, and it's based on the energy. And if you can read energy, you are being guided. If you can't read energy, you don't know what's going to happen next. If you follow the words, that's where we have been sold a bill of goods, where people say, yeah, these are good words, what do you think? And then I'll buy that because the words. And I say, yeah, but if you ever stop long enough to say, how do you feel about it, not what you hear about it. It empowers you beyond anything in the world. So the wake-up call now is, yeah, listen to people, fine, but feel it, people. Feel where you are. Feel, is this what I want to do? Do I want to do this or I want to do that? I say, you can ask your head and rationalize anything. But I say, don't ask the question of your head. Ask the question of your heart. Your heart is the monitor of the vibration. Your heart will give you an answer more accurate about anything in your world than your rational thinking will.